Hi, welcome back to CS372. Today is lecture 15. We cover where is consciousness. If you look at the roadmap of our lectures, and we are in the final lecture series of the quarter on consciousness and the mind. And this is the third lecture. And the last lecture, I covered what is consciousness. And I first motivated why did we study this particular subject. The reason was because our current AI hits some limitations, including generalization problem, and we cannot explain causality and so on and so forth. Therefore, like Yoshi Benjo was saying, we need to start to study system 2. And system 2 is pretty much depicting consciousness. Therefore, we can model human thinking, and we think logically, sequentially, and we do planning and reasoning. Okay, and we have been arguing whether system 2 or consciousness is computation all the way, and some scholars consider so, and some scholars do not consider the same. And when we get into where is consciousness, hopefully there will be more evidence to support one or the other. So let's quickly review what is consciousness. And there were many definitions, but I consider the ones who can be supported by science, and it can be more promising. And in particular, the proposal made by Kaku, a physicist in NYU, and he proposed consciousness need to consider time and space, which are evident, and also the number of feedback loops. And the feedback loop means sensing and the response. And flowers may have just limited number of feedback loops, including temperature and the water. And for pets at home, and the cats and dogs, they may have feedback loops for threats and for their hunger or interactions with their masters. And the human has tons of feedback loops. We have 5, 6 sensing organs. We have our mouth, eyes, and ears, skin, sensing sensations all the time. And when this energy level reaches a certain threshold, as Schrodinger said, we will transition into the consciousness state to try to handle these kind of challenges. And we have emotion, and other people have emotion. We conduct learning, and we receive feedback on our learning progress. So many feedbacks that makes human being the most sophisticated being on Earth. So quickly review this stimulus response diagram. And we started from stimulus and to human being, and started from the sensory circuits, and we can perceive sights, and we can hear sounds, smell, odor, or fragrance, and taste different food, so on and so forth. And these sensory qualities, as we discussed last time, will be brought to our nervous system, our brain. Our brain consists of billions of neurons divided roughly into 2 parts. Our cerebellum manages unconsciousness, and our cerebrum manages consciousness. And eventually consciousness or unconscious state make a decision on the corresponding responses to the stimulus, and then we finish this pathway. And another definition or sort of like discussion made by physicists is made by Erwin Schrodinger. And I found his What is Life book to be really intriguing. And he talked about evolution. He considered consciousness has something to do with learning and adaptation. And physically, the transition between consciousness and unconsciousness can be explained by physics laws. For the human state to transition from unconscious to consciousness, and Schrodinger says it's pretty similar to quantum jumps, when atoms could be in different states, and only when the energy reaches a certain threshold, and the quantum jump may occur or occur with a probability. And likewise, our sensing organs, let's say our skin, we sense some itchiness but not severe. It doesn't really trigger a response, but only the itchiness reaches a certain threshold, our consciousness aware about the itchiness, and we start scratching our skin. Okay? And so, but on the other hand, we talk about transition from consciousness to unconsciousness. And Schrodinger uses the second law of thermodynamics to depict the phenomenon. And the second law says in a closed system, and the entropy in a closed system will continue to increase until the entropy is maximized. And when that happens, the system is in an inert state. So the example I gave a few times was like driving, or we learn how to walk, right? Initially, we need to really pay attention to these activities in a learning mode, but eventually they became a routine, habitual to us, and they will fade into unconscious state. And only until some unexpected event happens, for instance, unexpected pedestrian shows up in front of us, then we are brought back to the conscious state to deal with this kind of novel challenge. We have to plan to react, to try not to hurt this pedestrian. So observations, we talk about the transitions, we talk about needs and wants, and we will expand the discussion shortly. And then we last time talked about top-down attention is in consciousness, and bottom-up is from unconsciousness to consciousness. And finally, unconsciousness is loaded with inherited skills, right? When we were born, and we already can do very sophisticated organ operations. Even let's say you ask us to start to learn how to operate our heartbeats or changing heartbeat rhythms, I think it probably cannot be learned. So the tough skills we inherited through DNA. And at the same time, all the learned skills on the earth, like we learn how to bike, swim, they never get written back to the DNA. So we don't pass the DNAs of swimming to our children. They just have to relearn everything we learned from afresh. So this is rather interesting and ironic. And this is touching the notion I talk about wants and the needs, right? It seems to be, I just briefly discuss it here. It seems to be what is essential, what are essential or what are the needs, respond to needs, will pass through DNA to our offspring. But what are optional, like our wants to have a new iPhone, our wants to be happy living in a better house, and those are really not passing along through the DNA. And if we look at another problem, unconscious to conscious, last time we didn't talk about it, was unconsciousness, our different organs or sensory circuits are collecting information simultaneously, right? Our eyes, our nose, and the skin, they collect respective signals at the same time. But only one consciousness at one time. So in other words, the consciousness is working single-threaded. And so the question here are actually twofold. One is this kind of limitation of human? We can only do one thing at a time. We only have one self. We cannot have multiple selves or multiple conscious existing at the same time. If this is a limitation to a human, suppose we want to build a robot. Do you think a robot can have multiple consciousness at the same time? So this is your homework number 1. The assignment will be given to you at the end of today. We have 2 questions about cancer and 2 questions about consciousness. So the first question here is why one self and one consciousness? My own hints are the following. And I remember in the 90s, I worked on the first generation of multiprocessor at DEC. Right? Even let's say we started with 4 CPUs, and we need to have this called spin lock on the top to try to synchronize those CPUs. And you can consider it's like a schedule on the top. And from the notion of writing to the memory or writing to the file, we always want to be able to ensure consistency, right? Consistency means there is an earthly order of events. You write something, you read something. If the order get messed up, then inconsistency may happen. Let's say you withdraw certain money, then you pay for certain merchants. If the order is out of sync, then someone will lose money. And so even simultaneously, many things can be going on in the background, but there should be one central scheduler. So even multiprocessor situation, we have to do synchronization. And the second notion is human may be constrained by our physical situation because we only have one body, right? We only have one pair of eyes. We have 2 hands and 2 legs. And even we say our sensation on the left-hand side and on the right-hand side, and both are urgent, want us to act on some responses, but we can only do one thing at a time, right? And so we argue this could be the limitation of one body of a person. When we implement on a robot, do we have the same constraint? So this is a question I think encourage you to discuss and express your thoughts. Okay. So where is consciousness? And Carl Desirath, and today pretty much we're presenting his findings, his research results, and he considered knowing where is very important. So just like everyone, we agree we live in this earth. We have this 3D Cartesian space, and we have the notion of time, right? If we don't have memory, then time may not exist, but memory is essential for us to do reasoning. So therefore, time is a byproduct of a memory. And then Desert talk about values, and values are important for human to evolve and for human to get rewards. So we do planning to get maximum rewards. But I'll come back to talk about value shortly, because the values, there are 2 sets of different values. One set of values is for survival. And even those could be brutal, right? Let's say I'm really hungry, or lion is very hungry. Lion has to hunt another animal. And that's the value of intrinsic value or needs of lion. And you may judge lion to be brutal, but from evolution perspective, it should be neutral. But from the moral perspective, we are not judging just needs, we're judging wants, right? You say, oh, you have no problem to survive, but you are stealing things from other people. Those are kind of distinct values. One set is intrinsic, the other set is sort of like for wants, different. Let's come back to visit those. But why do we need to know where is consciousness? And to be able to visualize consciousness, visualize the stimulus to neurons, and also responses generated by neurons, and we can do quite a few remarkable things. And in the next slide, we get to details. But let's talk about another mechanism discovered by Desirath in a very surprised manner. It's communication mechanism between neurons. And neuroscientists always thought neuron communication should be local. Consider our skin, we feel itchiness, right? And the itchiness, let's say, on my palm, And palm is very small, relatively small compared with my entire body, maybe 5% of my body. So the neurons get into communication of this itchiness shouldn't be very, very vast, right? We consider to be maybe okay, 5%, 10% of the body. But it turned out there is a broadcast effect. The broadcast effect means although sensations may be regional, but when the sensations is about to raise into the consciousness state, yes, it broadcasts to all the neurons. And other neurons may be managing other sensory circuits. The reason of doing so is because we only have one consciousness. And the consciousness is only single-threaded. And we cannot ask our consciousness to deal with two sensory circuits at the same time. Let's say walking to the left and running to the right. And broadcast seems to be a mechanism to just let everyone know the current situation, so some can back down a little bit and some can just move forward. In a sense, it's kind of prioritization based on the urgency or based on intrinsic values. Okay. Now, this is the book. This is the foundation of today's lecture, where is consciousness. And Carl Deseroth published his book last year. This is a distinguished book, not only in this scientific content, but also literature values. And as we talk about, which is also mentioned by Deseroth, and one, we want to visualize our brain. We want to find out causality, right? And the Deseroth team in around 2001 to 2005, they invented a new technology called Optogenetics. And I will talk about this method compared with the traditional electrode method, which one is probably more flexible, and why optogenetics is becoming the mainstream recently. And it's also critical to go back to revisit values. And intrinsic value is required by evolution, survival. And extrinsic one is optional, and then moral assessment will be involved. And that will be covered in the next lecture, ethics. And you may be surprised we will be able to quantify certain ethical behavior. Even we can quantify sins. Okay, let's defer until the next lecture. And for intrinsic values, and we have a set of mental disorders covered by Deseroth. And mental disorder, when we call disorder, means our needs, right, is going to one direction, but our mental reaction, mental response goes to the other direction or opposite direction. So one example is eating disorder, which will give tremendous treatment in this lecture, is our intrinsic value will say, oh, we have needs to eat when we are hungry, otherwise we will be dead, right? We are thirsty, we have to drink. But for eating disorder patient, they defy these fundamental needs. And the challenging issue for the psychiatrist is to find out why. If you don't know why, then the diagnosis is really, really ambiguous. And the treatment is virtually impossible. So before we get into more details, let's review how the brain structure works, and so get some foundations. And our brain consists of about 16 billion neurons, or probably even more. And the neurons interact with each other, connect to each other through synapses. Okay, and let's go into details to understand how it works, and then we can understand how these different visualization tools, why one is more effective than the other. And we see a neuron on the bottom left. The inputs to the neuron is a number of dendrites. And you can consider dendrites will be the branches of a tree or the root branches of a tree, which get nutrition from the soil. And likewise, the dendrites here, they are input ports getting signals, electrical signals or chemical signals from their upstream neurons. Can be thousands of neurons. And then the information eventually convert to electricity in the soma. Soma is the main body of a neuron. And then the electricity flows through the axon pipe and reaches eventually to axon terminal. The synonym of axon terminal is synaptic terminals. And you can consider that as an output port, right? And on the right-hand side, we see this how the input and output works in this synapse. So the output ports, axon terminals, emits either electricity or chemical. When the emission is in chemical, then there's a gap between the output and the input dendrites. But here we can kind of ignore the chemical transmission, because eventually when chemical reaches the other side, the next neuron or the downstream neurons, the signals will be converted to be electrical anyway, right? So we can, in general, thinking about only electrical information passing around between neurons.
Today, treatments to mental disorders only mitigate symptoms, not really addressing the causes, because we simply don't know the causes. And suppose we can collect neuron activities of a disorder, then both precision diagnosis and precision treatment will be possible. But how can we collect neuron activities given the disease? And we need to have visualization instruments. There have been two visualization technologies developed for visualizing neuron. The first one is Electrode, which was developed about a century ago. And the second one is Optogenetics. Electrode injects current into neuron. And there are two major metrics to measure the effectiveness of electrode technologies. One dimension is the temporal dynamics, how long the current can apply to neurons. The second one is the spatial resolution, how many neurons will be covered by this stimulation. Of course, we like to have fine-grained control or fine-grained stimulation, so therefore they call this resolution or the spot size should be as small as possible. And unfortunately, according to the survey of Home and the Lieber done in 2019, although the spot size can be further reduced, and this is really similar to Moore's law on the speed up of CPU, and the reduction of spatial resolution can also could be further reduced by about 10% every year. But the sensitivity of the neuron to electricity can be an issue. The duration of the exposure cannot be too long. And for monitoring a mental disease, and typically we need to cover the patient for days or even weeks. So therefore, maybe using light is less invasive. And indeed, using light, not only we can have better resolution and we can have longer temporal coverage, also the light can be able to reach deeper neurons without much trouble. And this optogenetic technology was developed by Carl Deseroth in early 2000. And later, the MIT professor Ed Boyden also joins the effort. And the basic technology is the following. First of all, it was motivated by the fact there are certain bacteria swimming underwater, and they will swim toward light. And once the light can shine on the bacteria, the light turns into electricity, and the bacteria gains their energy. So the same idea, if we can apply this kind of photosynthetic or photosensory molecules, DNA, into the neurons, then potentially we can shine lights on neuron, and the neuron will be kind of triggered. A neuron will generate electricity to the downstream neurons. And the light can then be used to turn on and turn off these neurons. And there can be some remarkable resolutions done. The first one is we can target individual neurons. The second one is we can target different types of neurons by doing gene therapy. And of course, there are limitations which we will cover in the end of the lecture. There has been some promises. There are several animal trials going on, and two clinical trials already ongoing, one for blindness treatment, the other one for retinal degeneration treatment. And following is a short video instructed by Boyden on this optogenetic technology. And let's take a listen.
The brain is made of maybe thousands of different kinds of cells called neurons that are built into very dense intermeshed networks that communicate. Each of these neurons computes using electricity. This cycle implements behavior and thought and emotion, all these different kinds of things. We also think that deficits in these electrical computations underlie many brain disorders that affect over 1 billion people around the world. In optogenetics, what we're doing is we're putting molecules that convert light into electricity into neurons, the cells of the brain. Then when we shine light on those neurons, light gets converted to electricity and allows us to turn on or off those cells. The goal here is to find a way to control the electrical activity in some cells and not others in the network. To do that, we had to turn to the natural world. It turns out that throughout all the kingdoms of life, in plants and funguses and bacteria and so on, you can find photosynthetic or photosensory molecules that convert light into electricity. So we borrowed these molecules from nature, and then using tricks from the field of gene therapy, we can put them into neurons. Now, these molecules can convert light to electricity, and they do it just in the neurons that we want to control, and not all their neighbors. So we can deliver these molecules to some cells and not others, and then we shine light on the network, we can turn on or off that subset of the cells. If we can turn on or off a set of cells that's embedded within this dense matrix, we can figure out how do they contribute to a behavior. For example, if we can turn on a set of cells, we can figure out what kinds of behaviors can it initiate. If we can turn off a set of cells, then we can delete it momentarily and figure out what it's necessary for. So by being able to dial in information into cells in the brain and to delete them, we can try to figure out how they contribute to networks and the behaviors and diseases that arise from brain computations. We can hunt down the exact set of cells that are contributing to a specific disease state, or which, when activated or shut down, will remedy that disease state. That's really important because right now a lot of drugs are developed that target molecules. But molecules are found throughout the brain. And in fact, many cells in the brain might be very molecularly similar to one another. If we can target circuits in the brain, though, we might be able to develop much more specific drugs. Imagine if we could hunt down the exact set of cells in the brain that, when activated, remedy a brain disorder. And then we can go in and look at the exact molecules in those cells. Maybe we can find drug targets that are much more specific than existing ones. You might also imagine that we can use optogenetics to directly control brain circuits in patients with brain disorders. Electricity is used to stimulate the brain in deep brain stimulation. If instead we could actually aim light at certain cells and turn them on or off, we might be much more specific. Rather than using electricity to turn on and off the cells of the brain and have many kinds of cells activated, the ones we want to affect as well as all their neighbors, if we can make just one disease-associated subset associated with light, and we can turn them on or off, we might be able to treat them with much more specificity.
Once we have collected massive amount of training data, which consists of neural activities and also the target diseases, and we can turn this data into a classification problem. The training data has S and Y. S consists of neurons, their locations, and their activities, temporal and spatial. And Y is the dependent disorders. Okay, so given a lot of data, then we can train a function F. And then remarkable things can be performed. The first part is a diagnosis. Given a set of unseen data, X, then we can say, well, what is the disease of this patient? And also, if a non-disease, we see a certain pattern X, then we know precisely which neurons need to be turned off. And this could be a much more precise treatment procedure developed for mitigating mental disorder. I have used the term disorder several times. When we say disorder, means something is not normal. And when we claim some behavior is abnormal, then we have to have standard showing normality. And the disorder can happen biologically or morally. And moral disorder could imply non-ethical or illegal behaviors. Here we don't talk about morality until the next lecture. And we focus on biological disorder. And according to renowned biologists, when we talk about, let's say, biological disorder, we have to talk in the evolutionary terms. And according to, for example, the quote from Peter Medawar, he said, for a biologist, the alternative to thinking in evolution terms is not to think at all. So let's revisit the term evolution. I think we heard about this term before. Some of us may have some misunderstanding. Now about evolution as related to genomics. Evolution refers to the process by which living organisms change over time through changes in the genome. Such evolutionary changes result from mutations that produce genomic variation, giving rise to individuals whose biological functions or physical traits are altered. The part that causes confusion is mutations. The question is first, is a mutation a random process, or a mutation has a mission to adapt to the environment? In other words, is mutation a process of passiveness, or mutation actively try to adapt an individual's biological functions and the physical traits to fit the changing environment? The fact is that the mutation itself is purely random. It is independent of the environment. And individuals that are better adapted tend to survive and produce more offsprings, while the less well adapted tend to die or produce fewer offspring. Let's use an example to illustrate the random process of mutation. Hundreds of years ago, a brown hill at the New Mexico state, the mice over there, the color was in light brown. And about 100 years ago, a volcano eruption happened over there, and the dark lava covered the land. Within about 30 years, almost all mice turned their color into dark, the same as the color of lava. So now, do you consider the mutation is targeted to be darker color, so therefore the new generation can survive better? The answer is not really. Mutation is purely random. 1 of 100 by chance will turn into dark, and the rest will be probably other colors or maintain to be light brown. And because the background of the hill becomes dark, so dark mice cannot be seen by the predators, by the eagles, so they can survive much better. So according to statistics, if the survival of dark mice is just 10% better than the brown mice, just after about 20 years, all the mice on the hill would turn into the dark color. And this is the power of just purely random mutation, and the selection is done by the environment. The mutation has no intelligence to target the mutation to fit the environment. Now we have evolution under our belt. We can come back to talking biological terms. So evolution teaches us the fittest survive. And it's a natural law. Survival of a species requires survival of the current generation and the reproduction of the future generations. And so for survival, we ask ourselves, is hunger a normal sensation? is eating a normal response to hunger? If we want to survive, then defying eating is not a normal behavior. Therefore, defying eating disorder is not a normal behavior. And using evolution to judge whether biologically defying hunger is normal and abnormal makes a lot of sense. So why we use eating disorder as an example to study neural visualization? It's because eating disorder is probably the most sophisticated disorder, and also it's the most deadly and costly. And there are about 9% of the world population suffer from some forms of eating disorder. As next, we would like to visualize which part of the brain manages hunger. And after we have this hunger sensation appears on our consciousness, and which part of our brain, which set of neurons defies hunger. If we can locate the neurons defying hunger, maybe we can find treatments by turning them off. In the middle of our brain, there's a structure called hypothalamus, which manages our survival functions. And if you look at the picture on the screen, it's an orange structure buried in the middle. And the functions it manages including releasing hormones, maintaining our daily physiological cycles, and controlling our appetite including thirst, hunger, and also managing our sleep. It also manages our sexual behavior, emotional responses, and body temperature. Again, all these are basic survival functions. And with optogenetics, we can successfully identify the neurons in this region, which manages each of these functions. So precisely for eating disorder, we can identify the neurons which manages appetite. And the neuroscientists already show they can successfully turn on or off a mice neurons for hunger. But here, turning on or off the neurons for managing appetite is not helpful for curing eating disorder. Because if we turn hunger off, just getting the situation worse, and keeping the hunger sensation up, the person is not going to eat anyway. So let's visit these competing forces that causes eating disorder. First of all, hypothalamus, which manages appetite, broadcasts the hunger sensation to all the neurons. And it's a conscious decision for the individual to defy the eating needs because of its value system. For the sake of beauty, the person saying not eating. And only the person with strong will would persist this defiance. And in time, this defiance is able to collect or recruit sufficient armies in other areas of the brain to join this resistance. And we call this army as the second self. The second self competing with the first self, constantly fighting for eating or not eating. Let us now making some observations. First of all, a patient with eating disorder was first convinced of that losing weight is positive and rewarding. Then the person turns defying hunger into a mission and a challenge. And neurons are then recruited to tackle these challenges. And there's a strong correlation between a top performer at school or at work with a person suffering from eating disorder, because both top performer and the eating disorder patient, they are goal-oriented, they are strong-willed, they can formulate plans, and they can persist to tackle the challenge. Through neuron visualization, we have two findings. The first finding is, although the structure of hypothalamus manages the hunger sensation, it broadcasts the hunger signal to all the neurons. The neurons that participate in defying hunger is only a small minority. And if we can somehow turn off these resistant neurons, we can perhaps cure eating disorder. And also for other mental conditions, like stress regulating brain area become larger in patients of depression and bipolar. We may be able to manipulate the neurons responsible for stress regulation to mitigate the illness. So now we understand mental disorders much better, and we appreciate these visualization tools which can let us identify each neuron's functionality, either positive or negative with respect to their biological function. And next we come back to our central mission. Where is consciousness? The question we impose in the middle. And fortunately, through visualization, neuroscientists they already are able to roughly identify the regions manages our consciousness. The method of visualization is the following. The first of all, we can assume the area manages consciousness can be constantly busy and can be task independently busy. And secondly, if a person is put into a meditation state, thinking nothing and be very calm, if still there's an area of neurons being active, then that particular area may be the managing neurons, and they could possibly be the regions managing consciousness. Neuroscientists have already identified two areas in the brain where consciousness are managed, or you can say consciousness resides. And the first area is in this blue rectangle area, which is our prefrontal cortex. And the second area is in the red circle. Its name is retrospinal cortex. These two cortexes work in concert to manage our consciousness. And the scientists right now continue conducting experiments on lab animals to see turning on or off neurons in these two areas, what will be the effect to the animal behaviors. Now we have located consciousness, and we like to probe further questions. The first question is consciousness computational only? And there was a debate between Roger Penrose and Yoshio Bengio. And Yoshio considers consciousness should be computation all the way. And Penrose agrees that our cerebellum, which manages on consciousness, is computational only. But he disagrees that our consciousness is merely computational. My question is, if it's not computational, then what is it? And we could further conduct visualization to see the neuron patterns in these two areas. For computation, there could be one unique neuron activity patterns. If the consciousness area also exhibits the same pattern, then yes, we can consider consciousness is only computational. But if there are other patterns emerges, then there is a possibility there are other activities going on. Then we can conduct further investigation. And for optogenetics, this technology is not without challenges. Before we can widely use the technology to be a therapeutic tool, and we need to tackle a list of obstacles listed on the slide. And I'm not going to get into details, and if you conduct research in the area, you can look into the further problems and the potential solutions. And finally, I'd like to challenge you guys with the discussion question number 2. We know evolution involves in satisfying our fundamental needs. Otherwise, a species would extinct, right? And for wants, and we consider those are optional. Those are luxuries. To preserve a species requires satisfying the needs managed by hypothalamus, including maintaining organ operations, maintaining our body temperature, balance body hormones, and managing eating, drinking, rest, and sleep. And my question is, how about the wants? So all wants should be optional. Is crying a need or crying a want? We know subjective values like having good grades should be a want, right? If we all have poor grades, a human species can still survive. But crying is an interesting trait. From the theory of evolution, crying does not help us to survive. But why crying is a common trait? So one hint is the notion of evolution bottleneck, which was explained by Dederoth as follows. About 70,000 years ago, there was a major volcano eruption occurred on Earth. And that eruption caused not only earthquake, tsunami, but also much ash, dust, and vapor in the air covered the sun for 6 years, and also disrupted the seasonal rain. So lots of people vanished during the disaster. And the Earth suffered from a huge reduction in population. And you could consider this is a bias sampling process. In the extreme case, let's think about this Noah Ark, right? And Noah just select a pair of animals for all the species. And the pair gets selected is a minority sample of the original species population. And this sampling is highly unrepresentative. So suppose you pick a pair which happen to have the trait of crying, and crying was not the popular traits of that particular species. And then we get into this so-called founder's effect. And this may be the reason we have some traits right now which are not really equipped for survival, and they are with us because of this bias selection process. The slide shows a bias sampling example. The population with a lot of white dots representing people without disease. But with the small samples, maybe the selected instances all suffer from disorders. So when we today talking about some people may have disorder and ourselves may be normal, and we have to be quite careful because the truth may be the opposite. This kind of founder effect should make us more humble. And further reading, I include two very good books. One is again Desiree's Projections and Roger Penrose's Emperor's New Mind. And Roger's book depicts his vision about what is consciousness and where is consciousness. In the next lecture, Dr. Duvari will come back to continue the coverage of psychiatry topic. And then the final lecture of the series, I will cover ethics, love, and the mind. My attempt is to quantify these three elements. So therefore, when we build a robot, we can build a robot with good ethics, loving, and rich mind. So thank you for attending today's lecture. Hopefully your project is in good progress, and I see you on Wednesday.